Hello and welcome to cell division. So this is the GCSE topics. Let's get on to the next one. So, okay, I'm going to start off with a bit of a sort of recap, so refresher on what is inside cells. We're going to division. So we know this nucleus the nucleus is inside plant and animal cells. Okay, so some plants I've forgotten when it comes to this sort of thing. These things occur in plants too, most of them we'll go through. So the nucleus contains chromosomes. So each gene is a length of DNA. So I'll put my pen onto the screen. So I'll turn the pen on to sort of make it a bit clearer, because a lot of people, I mean, when I first learned, I got a bit confused with this. Okay, what that actually meant by that. Okay, so this diagram shows chromosomes. I've got it's called a karyogram actually. We'll see one from an exam question a bit later. So these are chromosomes. Okay, so this is a pair of chromosomes. Make that clear. Pair of chromosomes. They're called chromosomes. The word chromo means colourful, and under a microscope, yeah, they're quite colourful, aren't they? That's actually what they mean, colourful. Okay, a pair of chromosomes. So each one is a chromosome. That's a pair. Often they're sort of aligned in pairs like this. Okay, pair of chromosomes, and a chromosome. Get another colour. Is made up. Just want to get the colour here. Here you go. So each colour. So I'm going to go into another chromosome and make it a bit clearer. So like a section here. Okay. Is called a gene. So a little bit of a chromosome or a section is called a gene. Okay, and a gene is made up of DNA. So like the whole thing I'm drawing now, the whole chromosome is actually made up of a coil of DNA. Okay, so the chromosome is made of a coil of DNA, and a little portion of that, okay, of the chromosome, a portion of the DNA is called a gene. So that's actually quite confusing when they sort of interchangeably talk about it, because it's kind of all the similar thing. Okay, so here it says each gene is a length of DNA. DNA is a long coiled molecule, so it's like a sort of slinky spring DNA, or like spiral pasta stuff. It's a little bit like that. They're a long coiled molecule that can unzip, so it can sort of like come apart and copy itself when a cell divides. The DNA carries genetic information. Okay, so we've got to see a chromosome consists of a string, this is like a portion or a part of genes. Okay, so there's lots of genes make up a chromosome. So a gene carries an instruction. It may determine the colour of your eye, shape of your nose. Okay, some go into things like height, which is also environmental as well. Okay, so, and all animals are chromosomes, and all plants have them as well, actually, because actually some of the genetic studies we could get asked about as well in exams were about plants and how they put their DNA to the next generation, because plants are selectively bred for certain characteristics, like to have more tomatoes on the plant or things like that. Okay, so they've also got genes. So I know people sort of think about humans and animals and things, but they often forget about the plants. Okay, so we're going to talk about cell division. So we've got some pictures here. So each cell has a cycle known as a cell cycle. So after each cycle, the cell divides into two cells. So at the moment, we're actually just talking about mitosis. So normal, everyday cell division. So talk about meiosis a bit later. I'm going to put that keyword at mitosis. So mitosis is for normal growth and development. That's what showed a baby going into an adult here. So normal growth and development. And this is to show wounds. Okay, so repair to these. So this is the platelets have obviously come along first of all, but then from the blood, that's what these little dots mean. These are platelets, but then the cells divide to repair. Because I hear things like scars don't go away, but they can do. I've seen them I had like a scar on my hand in it, and it kind of faded over time. So the cells will try and fade, and so yeah, it's not always there forever. Okay, they'll try and obviously repair as much as they can, the cells. Okay, so this is actually still mitosis. Okay, so normal growth and development, repair, I know I keep writing it, but it's mitosis. Okay. So you've got the cell cycle, so how long it spends in each phase. So we're going to be talking about this a bit more detail in a moment. So replication of DNA to form 2 copies of each chromosome. So when you saw that karyogram, it was actually, that cell is getting ready to divide. So, because it's actually made a copy. And synthesis of new subcellular structures, so it makes copies of everything in a cell, that means. So that's stage 1. Stage 2, mitosis, nucleus divides. Then stage 3, the actual cell divides. So actually this can take different times, different types of cell. So it's in rapidly growing, it only takes a few hours, but in adult animals, it can last for years. You know, we don't really need to grow very much. But yeah, with repair, we need to do. So repair will be quicker, but if we just sort of growth and like renewing cells, so that's obviously why you get older, you don't really grow as much, things like that, because they've reached their sort of final destination. Okay, so let's have a look at this. So it's comprehension. What is meant by the cell cycle? So we can go back. So the cell cycle is the cell divides into 2. So the time, to say time, because the cycle is all the time, taken for the cell to divide. Okay, why is the cell cycle needed? So this is growth and repair. How many cells are produced during mitosis? So we're going to talk about those in a little bit more. So 2. During which period of the cell does growth occur? So I need to go back. So what does it say? So replication, so 2. So stage 1. Explain why explain why stage 1 of the cell cycle is going to have a shorter duration developing embryos than adult humans. Okay, so obviously embryos are growing. So need to grow and develop. You'll see some exam questions a little bit more precise than that. Okay, in the way that there are. So this is actually a bit harder. So mitosis, so sort of kind of focus of this bit. So cell division, 2 and 3 of the cell cycle. The cells produced are called daughter cells and are genetically identical to each other. So mitosis produces genetically identical. So we've got clones, basically. So we've got a normal body cell. So I know we're just going to draw 4 just to make it easier, okay? Because obviously we do have more than that, but we just want to get the principle across. So the first stage of the cell cycle, a copy of each chromosome is made. So if it has 4 originally, and 4, which can be put into 2 pairs, because often they're put into pairs, yeah, because that each one is a copy of each other anyway. Okay, but you don't have to go to that too much of that at the moment. Okay, in the first stage of the cell cycle, a copy of each chromosome is made. Okay, so now we've got 8 in total. And then it's quite simple mitosis. The cell divides into 2 to form 2 daughter cells, each with a nucleus containing 4 chromosomes identical to the ones in the original parent cell. So we've just cloned it, okay? So a little bit more detail now. So DNA replication. Okay, so it's the longest stage of the cell cycle we saw in the pie chart. Cells grow bigger and increase their mass because they're copying. Okay, yeah, they replicate, copy their DNA to form 2 copies of each chromosome called chromatids, ready for cell division. Okay, so, and then it's just a little bit more in detail. I think it's called spindle fibers. Pull them apart. I know it's not often taught now at school. I thought these were called spindle fibers, which they line up and they pull them apart and you create 2 identical cells. Okay, because they line up when they get pulled apart. Okay, so a bit more detail. The two sets of chromosomes get pulled to different ends of the cell, so it's going to get ready to divide into two. They get pulled apart, and the nucleus begins to divide in two. And we get two identical cells. Okay, so a little bit more detail. Cytoplasm and cell membranes also divide. Eventually, when the cell membranes become completely detached, this is a process called cytokinesis. We finish with two daughter cells that are genetically identical. Okay, so this is some questions. I won't fill these ones out, okay, because they are comprehension. So you go replay the video back. This is a video, okay. So I'll let people do this. So it's step 1, 2, and 3. It's just what we just talked about. Okay, let me go on to the next thing. Okay, so going on to meiosis. Okay, meiosis. So meiosis, this right here, is used to create the gametes. It's spelled a bit weirdly, isn't it? Meiosis. It's kind of a funny word. So it only produces gametes. So sperm and egg cells. Okay, we've got pictures there. Okay, sperm and egg cells. So just this part here, because it's for use of sexual reproduction. Also in plants, and I keep saying that, so but plants also undergoes, okay, meiosis producing eggs and sperm here, okay? Meiosis can produce eggs and sperm also in plants. So you might know, yeah, the sperm is being in the pollen grains, okay? And they do have eggs. Quite similar, so yeah, we do have a common ancestor, us and plants, okay? So you can get asexual reproduction, which is actually undergone, just in case you get this, which is by cloning. So some animals can actually clone themselves. So in fact, they would use mitosis, but humans, plants, generally, we use definitely humans, we use meiosis to produce the cells. Right, okay, so let's go on to meiosis properly. Now, animals and plants make gametes, just sperm and egg. So you've got the parent cell. When we say parent cell, we don't mean it like the mum and dad, we mean the original cell, to call it. So chromosomes make identical copies of themselves. So you see there's a similarity here, what goes on in mitosis. So similar chromosomes pair up. Okay. So sections of DNA get swapped. This becomes a little bit different, doesn't it? Okay, some swapping goes on. Okay, so that's why they're going to be genetically different. So a pair of chromosomes divide. So they've got little bits of each one. And they don't just divide once, they divide twice. So you get 4 cells produced. Okay, so, and they're all genetically different to each other. Okay, so the difference is really just the DNA swapping, okay, and the 4 cells produced, it divides twice. Okay, so this is, remember, to make sperm or egg cells, depending on what one you have. Okay, is that if you think is that all the egg cells in a human, all sperm cells in a human are all genetically completely different. Okay, so if you are a twin or you've got twins in your family, it's not because they were the same, it's because once the egg was fertilized, it divided into like the same. It's not because the sperm or egg was identical. Okay, you could get brothers and sisters that look very alike, but they're not identical. Okay, so let's just think about it for a moment. So we've got, so a cell with all of its chromosomes is called, actually I'll put it at the top here, it'd be easy to compare this one to the bottom one, won't it? So the original cell is called diploid. So that means all of its chromosomes. So most cells are like this. So skin, muscle cells, okay, all sort of normal body cells, apart from the gametes, are diploid. And the only ones that are diploid, so these have half, they're called haploid. That's quite easy, half haploid. So they have half the original number, half of the chromosomes. Okay, half the chromosomes. So let's So haploid. So select the correct, so I'll leave that for the moment. Okay, so sort of pause it. So the differences between mitosis and meiosis, yes, it's only really two different differences. Thing is, in males, sperm is created all the time, okay, but in females, you're born with your eggs. So the meiosis occurred before you're born. You're actually born with the eggs in the ovaries, so no more get created. So that's quite interesting. So yeah, mitosis is going on in everyone's body to growth and repair, okay, but if you've got sperm, then meiosis is still occurring, okay. If you've got eggs, then your meiosis would have been actually done, okay. So select the correct words for each gap. New cells are needed to something, needed for growth. And so you can get rid of that one and replace worn out cells. That's true, can be replaced. The new cells must have the same something in them and as the originals. I'm going to go for genetic information. Okay, each cell has a something nucleus containing the chromosomes. Yeah, this isn't there's lots of gaps, isn't there? It's got to group together. Oh no, it should be genes grouped together on chromosomes. Yeah, sometimes you have to read the whole thing. Grouped together on chromosomes. Yeah, so it's a good idea for you to sort of draw out the chromosome and the genes. This type of cell division that produces identical cells is known as mitosis. Okay, so we know that. So let's go to this one. A scientist obtained a mass of 0.0062 nanograms of DNA from a diploid human cell. Calculate the mass of DNA the scientist should obtain from a haploid human cell. Okay, so this seems more difficult than it really is. So it says 0.02, so nano, let's think about, so 0.0062 times 10 to the minus 9 nano, that's grams. So what's that in picograms? Okay, so 1 nanogram is 1,000 picograms. Right, okay, so that's a diploid. So haploid is just going to be half. So let's divide by 2 first of all. 0.00, so 31 times 10 to the minus 9 grams. Okay, 1 nanogram is 1,000 picograms. So we need to divide that by 1,000. So what you can do, you do it on paper, but you can calculate this one. So okay, so you've got that. So that would be divided by 10, 100, 1,000. Okay, so 1 nanogram is 1,000 picograms. Let's make sure I got that right. Ah, no, wrong. Okay, so okay. So if you exchange rate, I put into grams there, didn't I? Oh, so it's easy to make mistakes in. This is actually not a hard question. So if one, so we've got, let's write in nanograms. Yep. So because I put in standard form, I'm a physicist. Okay, so nanograms. Okay, so 1 nanogram is 1,000 picograms. So actually, we need to times by 1,000. Okay, so we can move. So we can move. So 0.03 is 10, 0.3. So we're going to go to 3.1 picograms. Okay. So it says a student used the method shown in figure 14 to compare the mass of DNA extracted from strawberry fruit cells and from kiwi fruit cells. They crushed the fruit cells in a buffer solution, filtered to separate the soluble DNA from cell fragments, add ethanol. State why ethanol is used. So ethanol is used because DNA dissolves in it. The DNA dissolves in ethanol. State 2 variables a student needs to control when using this method to compare the mass of DNA from these 2 fruits. Oh, see, I missed that. Strawberry fruits and kiwi fruits. So it's 2 different fruits. Okay, so cell fragments. So you need to have the same mass of fruit and same amount of ethanol. Oh, amount. Okay, volume. We'll be more precise here. I wouldn't use amount of volume, sorry. Well, the student repeated the experiment, I'd say, to confirm results or to make an average. Just 1 mark. Okay. Okay, mitosis and meiosis are processes that produce new cells. Compare the outcomes of mitosis and meiosis. Aha, this one gets asked a lot. So in 2 papers. Okay, so mitosis, saw that earlier. The outcome, so what's produced? So 2 identical daughter cells are produced. I'd put in there genetically actually as well. Identical daughter cells are produced. That gives us 2 marks. 2 gives us 1. Genetically identical gives us 2. Okay, meiosis. 4 genetically different daughter cells are produced. Okay, that gives us 3 marks. Okay, a karyogram, as I promised you earlier, a karyogram is a picture of the chromosomes found in the nucleus of a single organism or single cell. Figure 8 shows a human karyogram. Okay, so you've got 23 pairs. State 2 reasons why this karyogram cannot be from a gamete sex cell. All right, so we know that because that's the full amount, isn't it? 23 pairs, so it is diploid. Okay, so this cell is diploid. So it has a full amount of chromosomes. Also, look at this one. This is to determine what sex a human would be, male or female. For X and Y, so it is male. So it has, so the cell has X and Y on the 23rd pair. If it was XX, it'll be female. Male. On the 23rd pair, if it was a gamete, it would have only have X or Y. So egg cells, okay, carry X, and sperm cells, okay, sperm cells can actually be, they can either carry another X or a Y. Okay, and that's why If this fertilised this egg, we'd get XX, you get a female. If this one fertilised the egg, we get XY male. So in humans, it's a male sperm that determines whether the baby is going to be a male or female. Some animals the other way around, it's the egg's different, but in humans, it's the male sperm that decides it. But thank you for listening, and good luck on your exams. Bye.